So that's a great anniversary. You should know it, I think. Okay. Good. And now let's continue. And who was the founder of our Academy of Sciences? Do you know who was the founder? Sorry?
Peter.
Sorry?
Peter the First.
Peter the First, but he created, he expressed his decision, right, to found Academy of Sciences, but he died, and the Academy of Sciences actually it was created after Schubert. But that was his decision. But first, and do you have an idea how many Russian academicians were in the initial Academy of Sciences? How many Russian academicians were the members of the initial Academy of Sciences? Do you have an idea? 100.
Maybe not many, maybe because we were
Not many, but
at the beginning,
tens.
like 10, yeah.
No,
About 30.
nobody was. Russian Academy of Sciences, there were no Russian academicians and Russian great scientists at that time, and therefore only academicians from Western countries, from Germany, mainly from Germany, France, and other countries were invited to be academicians in Russia. And there was someone, academician Euler. Do you know this name, Euler? He was a great mathematician.
He's the one who discovered, who proposed the diagram of Euler when
So who
proposed the diagram, Euler diagram. Is that Euler?
Yes, yes. And Euler was a great mathematician, but at the same time he was a physiologist. And he wrote to his son many years later, some years later, well, I advise you to enter the Russian Academy of Sciences, because in Russian, people, the authorities of Russia, they pay a lot of attention to the development of science, and they pay great salary. That's not the case for the Russian Academy of Sciences today. Okay, but
One moment, okay? I will
Okay,
put up the
good.
demonstration, I'm sorry.
good. Well, and I remember the celebration of the 275th anniversary of the Russian Academy of Sciences. There was a great assembly with the invitation of all the presidents of the academies from Europe, from the United States, etc. And there, I recall the report of the president of the French Academy of Sciences, that was Gregory Monago, who was originally from Russia, in fact, from Moscow. He was 5 years old when her parents left our country. Well, so, and she told that that you should be very happy to celebrate this anniversary. And we have done the same in our country, but that was 150 years ago. Well, so, the Western academies in France, in England, in Germany, they were created more than 100 years before the Russian Academy of Sciences. Well, so now let's come to our person anyway, and our lecture. What will move to the methodology and advanced techniques for brain research. There was a short tale, so we will finish this lecture in several minutes, and then we will continue our course where studying cellular and molecular mechanisms of neuroplasticity. But first, according to our tradition, could you remind me what are the main principles of the methodology in studying in brain research? First, what I mean, what are the levels of the body, of the brain, which are studied by different techniques? Sorry? A little bit louder for all the people, because that's very useful, I think, for not for me, because I know it exactly.
Zijn aanwezig en non-aanwezig.
Huh?
Aanwezig en non-aanwezig.
No, that's well. So there are several levels for the research of the brain function. Yeah, yeah. You're welcome.
Maybe it's about the isolated neurons and the neural networks and functions in this kind of thing.
Well, so first, this is the individual neurons, which are selected, which are distributed, and they're collected in so-called tissue culture. We start with individual neurons. This is the first level. What is the second level?
Neural interactions.
No, then we study interactions, interneuronal interactions, in slices, in things, freshly prepared sections of the brain. And the last level,
Tissue.
The whole brain.
this is the
Whole
whole
brain.
brain, exactly. The whole brain. There are three levels. Now, if we take individual neuron, there are several levels of the neural functions in terms of specific functions, neurotransmissions, for instance. What are these levels?
Micro level.
Sorry?
Type of the level.
Well, so look, if the neuron is stimulated, is stimulated for secretion of a neurotransmitter, what is the first reaction of the neuron? At what level?
Or you mean the micro level? I don't really get it.
Well, so there are several levels of the interactive levels of the function of the neuron. The first level, this is the gene expression. Yeah? If we stimulate our neurons, and it should increase the secretion of neurotransmitter. First, it should increase the synthesis of neurotransmitter. And that means that the first reaction is a change in the gene expression of neuropeptides themselves or the enzymes of monoamine synthesis. You remember that? During this particular day, you should recall everything.
So first one is
The next level.
so first one is gene expression, and then afterwards is the chemical.
And then
The need near fever.
gene expression after that. Thus, according to gene expression, what is the product of the gene expression? The synthesis of what substance? No.
Encapsulation of the neurotransmitter.
And the RNA. Huh?
Neurotransmitter gets encapsulated in
No.
vessels.
The first, the product of the gene expression is the messenger RNA. Well, so that messenger RNA is used as a template for a synthesis of what?
Protein.
Protein, exactly. For proteins. And then some proteins, they play a role of a neurotransmitter, so small sigma systems. Or some of them play a role of enzymes for monoamine synthesis. And therefore, in this case, there is the third level of regulation of monoamine synthesis. at the level of the enzymes. What does it mean? This is the regulation of the enzymatic activity of these enzymes. Because the same molecule of the enzyme could transfer either 10 molecules for a second, or 100 molecules, or 1,000 molecules, and it depends on the activity of the enzyme. Clear? Huh? Huh?
So first,
Lomonosov was more intelligent.
so on the first level, what happens with the RNA again? Because I get that on the second level, RNA is used to kind of synthesize proteins.
Yeah, is it?
But on the first level, what exactly happens to the RNA?
Well, so first, I repeat once again. The reaction of the neurons is started from the gene expression. What does it mean? That the DNA is dissociated, double strand DNA is dissociated for two strands, and one of the strands serves as a template for a synthesis of messenger RNA. Okay? One strand messenger RNA. Okay? And it happens in the nucleus, in the neuronal nucleus. Okay? And messenger RNA is transported to the cytoplasm. It leaves the nucleus and it enters the cytoplasm. Okay? And in the cytoplasm, messenger RNA serves as a template for protein synthesis from amino acids. Understand? Good. Well, and
But can you please say what happens to the other RNA? It just stays still and waits its turn? Or?
well, so messenger RNA. Well, so there is a short time, short-term life of all the molecules of messenger RNA and of proteins, because these molecules are degraded enzymatically by messenger RNases. This is enzymes which degrade the messenger RNA. Peptidases, the enzymes which degrade proteins. And monoamine oxidase, the enzyme which degrades what? Don't sleep. Monoamine oxidase, it degrades what kinds of molecules?
Monoamines.
Monoamines. Okay, good. Good morning. All of you. Good. Okay, it's clear? Okay, let's continue. Well, sure, and we'll. The last point of our last lecture was the evaluation of so-called non-invasive techniques for brain research that is particularly important for the study of human brain in norm and pathology. And you should remember that there are two kinds of non-invasive techniques, most important, fundamental non-invasive techniques, which are complementary. Could you restore me? Could you remind me what are these techniques? Two kinds of non-invasive techniques for brain research.
MRI and PET.
Good, good. MRI and positron emission tomography. Why they're complementary? Because we will turn actually to positron emission tomography. We discussed a lot about MRI during our last lecture, and now we will turn to positron emission tomography. But I mentioned already why these techniques are complementary.
Why? Because they focus on different things. Because when we use it for MRI, we measure the volt signal, the oxygenation on the blood and the brain, right? And when we're using PET, as I understand, we use the contrasting liquid or contrasting medicine of some kind, and we try to identify the expression of neurotransmitters in the brain, so they focus on different things.
Not really. Because these techniques are complementary by their resolution capacity. Because with the MRI, we could study first, we could study the morphology, the structural organization of the brain with a high resolution and 3D reconstruction, okay, at a morphological level. And with spectroscopy, we could differentiate only some individual elements like iron in tissue, etc. But at the PET, why PET scan is complementary? Because in PET scan, it has a much higher resolution. And we could study with PET scan, we could study molecular mechanism. This is morphological study of the brain, of the brain as a whole or in small regions of the brain, MRI. But PET scan, these are the molecular processes which are related to individual specific neurons. Huh? Okay? The difference. Understand what is the difference?
So why I was wrong?
Sorry?
I didn't get why I was wrong because that's when you're limiting expression of spirit molecule that.
But,
And
and I will explain you. Because we didn't study. What does it mean? I checked it, but it was functional.
I guess you have to sorry.
No, it works.
На трубку один раз, там установился, там видео играет, ну теперь там видео.
И что надо закрыть мою презентацию?
Нет, все, теперь должна работать.
Да.
Работаешь?
Не знаю.
Okay, okay. What about PET scan? Huh? Because we have discussed last time, we have discussed only MRI. Look, please, what is it? If we study the molecular mechanism, we should have a facility, an instrument, to label specific molecules, functional molecules, isotopic ligands. Okay? Yeah. So if we deal with dopaminergic neurons, as it is demonstrated here, what do you think? What are the most important functional molecules in dopaminergic neurons? What are the fundamental functional molecules in dopaminergic neurons which are responsible for dopamine synthesis, for dopamine uptake, and for dopamine intravesicular storing?
L-dopa?
L-dopa, this is an immediate precursor of dopamine. Good, that's okay.
Tyrosine?
Tyrosine, not tyrosine, because if we use tyrosine, this is not a specific functional molecule of the dopaminergic neurons, because L-tyrosine is used for the synthesis of most proteins all through the brain and the whole body. What are other molecules? Enzymes. Dopamine synthesis. Do you remember how they are called?
Tyrosine hydroxylase?
Tyrosine hydroxylase, the first one. The second one. Tyrosine hydroxylase is a specific one. The second is non-specific. How it is called?
It's not L-dopa?
No, L-dopa, this is a precursor.
Yeah, okay.
This is a synthesis. The second enzyme is
The aromatic.
aromatic L-amino acid carboxylase. Good. Huh? Then, what is a functional molecules which is typical, is characteristic of dopaminergic neurons and for monoaminergic neurons in general, but which does not exist in peptidergic neurons. The only one molecule, which is very specific only for monoaminergic neurons.
Monoaminoxidase?
No, this is an enzyme for degradation. Yeah, monoaminoxidase, good. That's okay. That's all. But what is another molecule? Even more specific. Because there are there is a kind of molecule which is specific for all monoaminergic neurons, but which are also specific individually for dopaminergic neurons, for serotoninergic neurons, and for norepinephrine-nergic neurons.
Is it somehow connected with the reactive?
Yes, exactly. These are monoamine transporter. Right? Also, monoamine oxidase. It is specific for all monoaminergic neurons. It degrades all monoamines, dopamine, serotonin, and norepinephrine. While there are absolutely specific molecules like dopamine transporters in dopamine neurons, serotonin transporter in serotoninergic neurons, and also norepinephrine transporter in noradrenergic neurons. These are absolutely specific. Well, and in this picture, you will see the axonal terminals of dopaminergic neurons, which impart on the target neurons with the receptors for dopamine. Okay. Well, so first we could use fluorodopa, isotope of dopa, to study with PET scan. We can use a dopa to study the speed, the rate of dopamine synthesis, or the activity of aromatic amino acid decarboxylase. Okay. Then we have a ligand for B2 receptor for receptors for dopaminergic neurons, and there are also ligands for it is not drawn here for vesicular monoamine transporter type 2. Well, and now look, first, with fluorodopa, the repeat 1A, once again, we could study the rate of dopamine synthesis in the striatum. This is striatum, look. Well, and this is striatum of a healthy guy, and this is striatum of a patient with Parkinson's disease. Look, the intensity of inclusion of fluorodopa is significantly higher in healthy people than in ill people. Okay. And also there is a sort of asymmetry. Look, from this side, this perforation is weaker than from that side. This is typical for Parkinson's disease. But I will explain to you why we need to study the number of these receptors for dopamine. Because there is a sort of upregulation between the dopamine release from presynaptic terminals and the number of V2 receptors. What does it mean? If there is a lot of dopamine in the intercellular cleft, the number of receptors is very low. You understand? And on the contrary, if there is only traces, small amount of dopamine in the intercellular cleft, the number of these receptors increased significantly. And this is a sort of adaptive reaction. Well, so in the case of a low level secretion of dopamine, the target neurons increase their sensitivity because the number of D2 receptors, this is a characteristic of the neuron sensitivity. Well, so
So in the neuron, the receptors can, the amount of receptors that increase and decrease respectively.
Exactly. And it depends on the amount of dopamine in the intercellular cleft.
How long does it take to increase or decrease number of receptors of neuron?
I think it's seconds or even less than seconds, because these receptor molecules, they should not be synthesized de novo, because there is a lot of protein of these receptors in the cytoplasm of the neurons. And if there is a need of a lot of receptors in the membrane, these receptor molecules, which are contained in the cytoplasm, they enter the membrane. Okay, and these are seconds, really seconds. Well, and what is the practical application of this study of these ligands for receptors? Let's evaluate the level of the expression of these receptors. We could consider what is the rate of dopamine synthesis. You understand? If there is a lot of molecules of receptors, we know that there is a low level of dopamine release. Right? If there are only few receptor molecules, we know that the level of dopamine release is very high. Huh? You understand? Good. Now let's continue. Well, and I mentioned that we use different models to study the brain at the level of individual neurons, interneuronal interactions in slices, and the whole brain. But on the other hand, there are only different facilities to facilitate the study of the brain by using very simple or complex of the organism with a simple or complex organization of the brain. For instance, in invertebrates, the central nervous system is consisted only of a few neurons. Only a few neurons. Huh? There's less than 10 neurons. And it is very easy and very useful to study the functioning of these neurons and their interaction in these invertebrates. There's not a lot of zebrafish, and also these organisms that are highly reproducible. Okay? So in rodents, the brain is organized in a much more complex way, and in non-human primates, but still, what is interesting, that all over the world, if we develop some medicines, some drugs, we should pass through two-step investigations. Preclinical investigations in animals, and then clinical trials in humans. Okay? And what is surprising, that we translate our knowledge about the mechanism of action of these medicines in rodents to human beings. And this is not correct at all, because we could translate this data, for instance, if we study the pharmacological action of the heart, because the heart is organized more or less in the same way, in the same fashion in rodents and in humans, but not the brain. I think you believe that the brain of the rodents is not the same as the brain of humans. Well, so that's very important, but it's not used, unfortunately. It's very important to use the intermediate model between the rodents and humans, non-human primates. Well, but this is a very expensive study. There are much ethical problems with the use of monkeys, and therefore, so far, they're not used almost. I tried many times to develop these studies, these preclinical studies in monkeys, in collaboration with other countries, with Taiwan, with Vietnam, with China, the countries where there's a lot of monkeys. But still, I didn't succeed for technical, political, or administrative reasons. Yeah, that's possible. Yeah, that would be excellent. Because even in Western countries, in Northern America and Canada, etc., the monkeys, their cost is extremely high. It's more than from $3,000 to $5,000 per monkey. Huh? Well, that was very interesting that when I visited Taiwan, these monkeys are developed in the closed military zone. Also, and I visited this military zone. It was very interesting when they are looking at the monkeys' families, which are walking in this space. Yeah, you know.
You said the problem, one of the problems of the time that in center for dermatology research, we told us that it is prohibited to use human-like monkeys for donation all over the world. Like a crime.
No, that's not really the truth, but you know, but there are some ethical regulations with the use of monkeys. First, we cannot kill monkeys. We cannot kill monkeys. And therefore, also in this particular case, for instance, with Parkinson's disease, there is no limitation to produce a Parkinson's disease model in monkeys by giving small doses of neurotoxin, specific neurotoxin or dopamine agonists. But we have no right to kill this thing, and therefore we should use it for a long time by treating with medicines, by starting with non-invasive techniques, the molecular targets for the diagnosis, for the treatment, etc., etc. Well, so that is not. These studies are not prohibited, but there is a certain limitation, ethical limitation, and we should follow them. But if we use monkeys as an intermediate stage between rodents and humans, certainly we should get enough information in rodents, and then we should check only fundamental points in monkeys. Not all these mechanisms, but the really fundamental points. Well, so that it means that we don't need a lot of monkeys, and we have no right to overcome these ethical rules. Okay, understand? Well, so that's a good idea to try to develop with your country. Why not? Well, experimental approaches. Well, what are the experimental approaches for the in vivo studies in the whole animals? Today, this is a pharmacology. We can use agonists and antagonists of principal functional molecules. Do you know what does mean agonist or antagonist? Well, so agonist, for instance, if we use dopamine, if we start with the functional activity of dopamine and functional neurodopamine, dopamine itself is not a stable molecule. And therefore, we should use another molecule which reproduces the same physiological effect as dopamine, but which is stable. Okay, this is agonist. Usually, we say agonist if we deal with receptors. Well, so this is the substances which affect certain receptors. And antagonist, this is just on the contrary. If we have no idea to stimulate the receptors, but on the contrary, we have an idea to inhibit these receptors. Well, we should use antagonists. These are inhibitors, first of all, of receptors, specific receptors. Well, so there is a great pharmacological kitchen, actually, with the use of agonists and antagonists of principal functional molecules, of neuropeptides, of monoamines, etc. Then, the modification of specific genes by using knockout, knock-in, overexpression. What does it mean? That we mentioned already that all the physiological processes, all the physiological synthesis are started with gene expression, okay? And proteins are synthesized on messenger RNA as a template, okay? Well, so if we have a tool actually, if I'm interested, we could cut certain genes. If we study the role of a certain protein in the function of the neurons, first we could eliminate these genes in animals. Then, if we study the role of another gene, we could, on the contrary, we could implant or another gene or a gene with a high activity, with a higher level of expression. So, and this approach is called overexpression. Okay, and well, so these tools are widely distributed all over the world. Unfortunately, in our country, we have a weak point with the use of these animals. Because these animals are produced actually in Western countries commercially, and they are sold. But there are only a few lines of these animals with knockout. I'm happy to have a line of transgenic mice which we bought in Japan. And this is very interesting line. Well, well, since we're interested in dopaminergic neurons, they're very interested to visualize, to detect always, to be visible, tyrosine hydroxylase, the first enzyme. And these animals, the genetic program of these animals, transgenic animals, is modified, and with the gene of tyrosine hydroxylase, there is an expression of so-called green fluorescent protein. This is a protein which has its own labeling with a fluorescent stain. You understand? Well, so if there is an expression of tyrosine hydroxylase, we cannot see it, we cannot visualize it. But at the same time, these animals produce the protein which is labeled by fluorescent stain label. And therefore, if we see the neurons which are stained with green fluorescent proteins, we know that these neurons express tyrosine hydroxylase. Not clear?
No.
No?
No.
Not clear. Look, Wilson. There are genes. There is a set of genes. And there is a gene for tyrosine hydroxylase. And tyrosine hydroxylase is, apart from this gene, tyrosine hydroxylase is synthesized as protein in the cytoplasm. But in this case, we cannot see, it is not visible, this tyrosine hydroxylase in the neurons. In order to make this synthesis visible, we introduce in the vicinity another gene, which is called green fluorescent protein. The gene of another protein, which has no functional role, but it is stained already. Okay? And then these neurons, in addition to tyrosine hydroxylase, there is a synthesis of this green fluorescent protein. And therefore, in this neuron, we can see it is visible in the neurons. And if we detect gene fluorescent protein, if we see this protein, this fluorescence, we know that these neurons at the same time express tyrosine hydroxylase.
Is this like a weird thing? Because I was reading that in order to see things in the telescope, you usually use extract materials like coloring materials, like, metals and stuff.
And so if you can use something that is already inside the body to color, that is a rare thing, or is it more often used than when use like materials, metal or stuff?
Look, you're right. That if having known this GFT, we could detect terrorism encapsulates by its specific labeling with antibodies labeled, stained with the fluorescent marker. It is possible. But still it takes time, it takes money, okay? And we cannot do it for a lot of animals. This labeling is available for a lot of animals in a large experience, particularly if we do preclinical studies for testing medicine, drugs. We should use hundreds of animals. And therefore, and in this case, we do not need to stain anything. Good. Okay, you're right. Yeah, please.
Yes, I also have a question about this. Could it be the case that the gene that is that produces GFP is active, while the gene that is responsible for terrorism encapsulates is not active? I mean, we cannot see it, so is it possible, or they are always both active? Because some genes might be
Always. If we see GFP, fluorescent protein, and fluorescence of the neurons, we know exactly that there is an expression of tyrosine hydroxylase, of gene expression. It doesn't mean that there is, well, sure, we are sure that there is an expression of gene, and therefore there is a synthesis of messenger RNA. But we can say nothing about the amount of the protein of tyrosine hydroxylase in the neurons, okay? And we have, in our experiments, we met a situation with a high level of GFP expression. High level, that all the neurons are fired, okay? But there is almost no protein, just traces of the protein. Yeah.
So if I understood you correctly, we can replace a gene that helps to produce GFP as GFP is a protein, right?
Yes, so,
Okay, fine.
Yeah.
I just wanted to clarify.
yeah,
I
that's right.
also wanted to ask a question about the difference of knockout and knockdown methodology. If I understand it correctly, knockout, as you said, is like to simply cut
Yeah.
the gene.
Okay.
And knockdown is just to make it like switching the color or something.
Exactly. Well, so initially, the technique for knockout was developed. It means that at the very beginning, at the level of embryos, very early embryos, we cut this gene, and after all, the whole organism is developed without this gene and without this protein. And there is a weak point of these animals because during their development with a certain protein, there is a certain adaptation for division of the whole body for the deficiency of this protein. No? Well, and therefore, people they developed another technique which is very similar, but it is called conventional or knockdown or conventional knockout. It means that we could, I don't go to more details, but in fact, we could cut the gene functionally. We could eliminate the functional gene during our experiment for a short time, for instance. And therefore, for this elimination of the gene, there is no adaptation. Okay. Well, so for instance, we're working in animal models using adult animals, and we can cut this gene for several hours, for several days in adulthood. Yeah. Okay. But this is not very easy, believe me. Huh? Okay, then let's go modification of specific electrical magnetic stimulation. I think that some of you use these techniques, no, in your study or in your laboratories. Huh? Electrical magnetic stimulation of the brain. If there is anybody? No, yes, okay. Will you tell a few words? Will you tell a few words about this technique?
Yes.
Magnetic stimulation.
Okay. So I'm for now focusing on electrical stimulation, cathode inhibiting say the prefrontal cortex. Yes. And it's kind of it doesn't elicit action potential or potential, but it transiently affects the membrane potential. So when an individual undertakes a task, then we can observe changes in that area.
Okay, good.
Yes.
Good. I've never used this technique in my experience, but I have an impression. I believe that these techniques there is a weak point that this action should be highly irradiated, diffusive, and it occupies a large area of the brain, and this area might be involved in the regulation of different functions. And therefore, in this case, usually there is a lot of side effects. But I think so, but I'm not really a specialist. But this is
Other things.
but this is the case for deep brain stimulation, because I mentioned probably many times that in the deep brain stimulation, in Parkinson's disease, there is an improvement of motor behavior on the one hand, but on the other hand, there are strong side effects, particularly depression and suicides. Well, okay, good. Thank you. Implantation into the brain of viral-based genes, stem cells, and genetically modified cells. Well, so this is a direction which is highly developed actually, and people believe that they will come to a good technique for the treatment of patients. What does it mean? What does it mean that genetically? Well, this is actually genetically modified cells. Well, so we could implant, in fact, if there is a deficiency of some sort of genes in the brain in patient, theoretically, we could inject the construction with the viral-based lentivirus, with the genes, with the necessary genes. Okay? And this construction is should be included immediately into the neurons and also in glial cells. Neurons are almost not reproducible, but on the contrary, glial cells are very highly reproducible, and therefore these genes will be reproduced, multiply reproduced, and very rapidly. Also, stem cells and genetically modified cells. I mentioned already, and I will attract your attention when we're saying about Parkinson's disease, I think in one or two lectures, that since the 90s, there was an idea to replace degenerated dopaminergic neurons by dopaminergic neurons taken, dissected from the healthy people. Well, but for 30 years, people tried to do it, and they reached an extremely good therapeutic effect in animal models, but not in humans, but not in patients at all. Well, so I will give you many times that I believe that there is a biological restriction. In other words, that the neurons which are implanted into the brain, they survive, they survive very well. They they provide their specific functions, the secretion of dopamine, but they are not inserted into the neuronal ensemble. And therefore, there is no regulation, at least there is no regulation of dopamine synthesis in these neurons. There is no feedback. The neurons do not know if they synthesize enough of dopamine or not. You see? And therefore, these neurons, implanted neurons, can be functionally integrated into the brain. They integrated structure. They reproduce their metabolic features, but they are not functionally integrated into the brain. And this is a biological restriction, I think. Well, and now, and these are different possibilities to use knockouts for genes, for example. If we deal with serotonin receptors, there are several types of knockouts for genes, for serotonin receptors, for serotonin membrane transporter, for vesicular membrane transporter, for enzymes of serotonin degradation, like monoamine oxidation. Okay? Well, so all the molecules, all the genes can be illuminated in this in animals for the study, specific study of these neurons.
Sorry, can you please clarify again about if we insert a neuron in a certain yes, in the area where dopamine is mostly generated. If I understand correctly, that neuron, it will generate the it will fire action potentials and neurotransmitters will go, but there will be a problem with I guess the volume aspect of connection between the other neurons, or what will be exactly the problem?
Look, this is a brain region, and there is a lot of neurons. Well, all the neurons are interacted in between, interacted, and they know, according to the feedback regulation, for instance, they know how much neurotransmitter the brain needs. Okay?
And the feedback regulation are the channels, or?
Yeah, the feedback regulations is provided by the autoreceptors, by the neurotransmitter at all. Do you remember I told you about autoreceptors, no? Well, so look, all the neurons, that was a topic of my lecture. Well, so all the neurons, they have a lot of receptors, dozens of hundreds of receptors for many chemicals, potential neurotransmitters or neuromodulators, for neuropeptides, for monoamines, etc., etc., etc. But there are also autoreceptors. And autoreceptors, they are sensitive to the neurotransmitter which is contained in the intercellular cleft. If there is too much these molecules, these autoreceptors provide a signal, send a signal to the neuron that it should decrease the production of this neurotransmitter.
It's increased synaptic neuron,
Look, there's no sense. Look, there are autoreceptors. For instance, you have autoreceptors. If I will touch these autoreceptors, okay, the body has a signal that you should save your head, okay, and this is the case for the neuron. There is an autoreceptors, and it reacts on the concentration of this neurotransmitter in the intercellular. If there is too many molecules, it sends the signal to these neurons that the synthesis should be decreased, okay? And on the contrary, if there is not enough molecules, it sends signals that it should be increased, okay? So if you, so in the brain itself, all the neurons, they're connected, they're interacted functionally in between with, including this feedback regulation. If you introduce another neuron, if you implant the neuron which was dissected from another brain, it will be developed, right? It will develop processes, actions, the drives, etc., etc. It will synthesize, produce neurotransmitter, but it will not reconstruct adequate relations with other neurons in this network. And therefore, it would remain functionally isolated from the brain. Okay?
But still, there will be connections. There will be processes that is connecting to other surrounding neurons.
Yeah.
There will be some connections, or there will be no connection.
Yeah, it could make a synaptic complex with other neurons, but with no sense as a healthy brain. Okay, this will not be let's say correct synapse. This will be just a mechanical synapse.
Then if it's not strong enough, then maybe synaptic pruning or something could take place and it will eventually die.
Also, fundamentally, I think that there is a sort of biological restriction that neuron do not know what neurons could be the target for this neuron, and what neurons should touch this neuron. Okay. So there is no adequate integration, and the neurons they have no capacity to organize specific synaptic complexes. Good example. Look, if we will take a certain brain region, dissect certain brain region, dissociate them into individual neurons, and we put it in tissue culture. Okay, different neurons. So our tissue culture, right? We dissociate a small piece of tissue, the neurons, and we will put it in the tissue culture. These neurons will develop processes, and these processes will establish synaptic contacts. But if you will take the same tissue, if you prepare the same tissue culture 100 times, in any case, there will be a different construction of interneuronal relations. Well, so there is a sort of regulation for the neurons to make certain functionally important relations, synaptic relations.
I think I have a hypothesis. Maybe all the neurons in the brain, they have something like ID, student ID. Mm-hmm. So when you bring in new neuron, it doesn't have an ID. So if we can find the ID for it and insert it, it will make a connection. Like first when I came to the class, I came late, so no one spoke to me.
Yeah, why not? Why not? I think that any hypothesis is very valuable, and it should be tested experimentally. But I always give you an example of this biological restriction. You think about transplantation of internal organs. Do you remember I told something about that? No. Well, so I recall this situation because for many years, for decades, the people they tried to implant internal organs, to replace internal organs, the heart, the liver, kidney, etc., etc. And they did not succeed to do it. And that was a surgical, actually, that was a surgical surgeon problems. And they conceded that they could succeed if they improve the technical surgical technique. And in our country was the most important, the most outstanding transplantologist. His name was Demikhov. Demikhov, he transplanted a lot of internal organs, mainly in dogs. Right? Well, and he said in other countries, people they transplanted these internal organs, and these internal organs they survived no more than for 2 days. But Demikhov was made a more sophisticated operation, and he succeeded to make these organs, implanted organs, surviving for a week. And therefore, he conceded that to solve this issue, he will be able to by improving his surgical techniques. But that was not the case, and it was clear when we have learned that there is a sort of immune rejection. Well, you know that immune rejection, and therefore any organ which is implanted should be rejected immunologically. And that was, and we learned it not far above. And therefore, but after the development of immunosuppressors, which suppress the activity of the immune system, researchers succeeded to implant and to keep this organ for years. Not really for a very, very long time, but anyway. But that was, I repeat once again, that this immunological rejection was a biological restriction. I think that this sort of biological, not immunological, but probably another one, biological restriction is the case also for the implantation of individual neurons. And I believe that one day you will be nominated as a Nobel Prize, you for instance, when solving this issue. Good luck to you.
But okay, so it won't get a good connection with other neurons, but if we take a neuron that used to synthesize peptides and put it in a place where it's mostly dopamine, will it synthesize dopamine? Will it learn to synthesize dopamine?
That's a good question.
No, that's for you.
I have no so much ambitions to be your coordinator. well, so that's a very good question. The question is, if a dissect neuron which produces neurotransmitters, for instance, or dopamine, in one brain region, and we put it in the region which is rich in other neurons, like serotonin neurons. Whether this neuron, implanted neurons, would modify its phenotype.
Yes, and will it forget?
Yes. It has been shown. It has been shown, and this is I will tell you a little bit later that this is one of the important phenomena of so-called neuroplasticity. Well, so it means, in fact, that the genetic program of the neuron is not conservative. It could be modified by so-called epigenetic factors. That's factors of the environment. And this, you mean the environmental factors around the neuron. Well, so these epigenetic factors could modify gene expression and the genomic program. Is it clear? You okay? Good. Well, so I think what are the weak points in our country? Well, so monkeys are not available for brain research because in our country, at the south of in South Finland, there is a station for the reproducing of monkeys. But these people, these monkeys, they cost great sum. They're not actually available for research. There are only few MRI scans and no PET scans for animal research. And this is true. So far, there are no PET scans for animal research in our country. For rodents, for monkeys, we can use PET scan, which is available for human beings. But that is not the case for rodents. This is a weak point. Well, now let's turn to our next lecture. I think that's one of the most intriguing characteristic of the brain. This is neuroplasticity. I mentioned already many times that we say usually, routinely, we say neuroplasticity, but practically we do not say cardioplasticity or kidney plasticity or something like this. Why? Because the brain, from the physiological point of view, brain is a coordinator, is a conductor of all the functions of the brain and of the whole body. And therefore it should be very, very flexible. Because even a small disorganization of the brain function would lead to a serious difficulties in the functioning of any other system, of cardiovascular system, of kidney, of mineral and water metabolism, etc., etc. And therefore, the brain is constructed by the God, and on that east, in fact, with a very, very high flexibility or very, very high plasticity. Now and there are two, and this neuroplasticity, it was declared, it was defined, it was intensely studied in the at the end of the 19th century, which was called as a golden century for experimental biology. At that time, they had no tools for the direct study of the molecular mechanism, of the cellular mechanism, etc. But they used, mainly the local destroying of the brain with the subsequent analysis of the functional consequences of this lesion of the brain. And Bexer was one of them. That was really a great guy, a great scientist in our science, and he lived at the same time as Pavlov, and they were Ivan Pavlov, Nobel Prize, Pavlov, but they were great enemies because they were absolutely different. They had absolutely different mind, different way of mind. Because Pavlov, he was really a scientist which was formed and which was formed as a Western scientist. Well, so Western scientists they declare small hypotheses, just small hypotheses. Then they provide proofs for this hypothesis, or they test this hypothesis, and only then they go further. Okay, well, step by step, small step by step. This is a style of Western partners. But Bercherev, he had just the opposite one. He was interested, like Lomonosov, he was interested in all the fields of neurology. He was a neurologist, he was a neuroanatomist, he was a neurophysiologist, he was a psychiatrist. Well, and he contributed a lot to all these areas. For instance, there is a brain structure which is called by name of Bercherev. And you know what happens with Bercherev during the Stalin's time? No? Well, so he was invited for a consultation of Stalin, and he diagnoses schizophrenia or something like this. Sorry?
The third stage of paranoia.
Yeah, parallel, yeah. Well, so, and after that, people believe that he was eliminated, physically eliminated. Picture effect. Well, so, and he was a very great scientist. As far as I know, it's not widely described, but as far as I know, he was nominated 3 times for a Nobel Prize. But the referee always was there. Europe was always Pavlov. And Pavlov always provided negative resolution. Well, so he did do it, yeah.
So, Baxter 3 times nominated for the Nobel Prize, and all the 3 times the Pavlov was there.
Yeah.
And what's the main problem with their conflict? Like, they're 2 great scientists. Okay, they have different approaches, but still, why do they?
You know that Pavlov is Western. Well, so I mentioned that Pavlov had a Western way of mind. Very punctual, very
Precise.
located, very precise, etc. And he considered that to all the people which are interested in different fields and they stay in one area 1 year and another one half an hour.
It's pathetic science.
That they are not serious researchers.
Ah,
That was an idea of Pavlov.
but Baxter made some important investigations. He cannot deny this.
Yeah. So Pavlov, he didn't believe that the people which are not focused on the same local area for a long time, very seriously, by using a lot of experiments, a lot of controls, they're not serious researchers at all, and therefore the people like Pavlov, they did not believe to the results and to the ideas of these people,
Okay.
like Dixon. So that was an idea. Well, and in fact, to a certain extent, I agree with Pavlov, and I know some examples, a number of examples of good researchers, which done something in parallel, and finally they did not reach good progress in any field. For instance, in our country, there is a very good researcher. He was a successor of Kashkayan's great name. His name of this researcher is Sakharov, and he's a professor in my institute. We are working together for 40 years in the same institute, but in different laboratories. But at the same time, he's very famous between. Do you know this word chansonnier? Huh? Chansonnier, which prepared the songs for like Nikitin and Hama. You know Nikitin?
Part.
Do you know Nikitin who prepared the chansons for? No, you don't know. Well, but anyway, he was a poet and he wrote songs and so. But finally, he did not succeed in research. He's still alive, but he's very old. But he didn't succeed, I think, because he did not focus enough and he was not concentrated into the research field. And there are many examples. Well, and probably you know the names. His granddaughter was academician Dekteva from St. Petersburg. Do you know this name? No? No. She was a neurophysiologist. And now he's how to say, his grandson son, he is also an academician in neurophysiology, but actually he is doing he's doing the physiology of yoga and something like this in staying in Nepal. Well, and let's return to the definition of neuroplasticity by Dexter. The function of damaged brain areas restored by the undamaged areas. This is due to reorganization of interneuronal connections in accordance with the information derived from the target zone. What is true, what is the fact, and what is the idea? What is true, the fact that that neurologists and psychiatrists they have demonstrated that some patients which with trauma of the brain, with local trauma of the brain or something like this, well, so they can restore their function. For instance, after the trauma, the people, they lose their capacity to speak, for instance. And some of them, with time, some of them, they restore this capacity. Or another, okay? Well, so this is a fact. This is experience of neurologists. The function of damaged brain areas restored, but that is restored. But what is the hypothesis? That it is restored by undamaged areas. This is due to a reorganization of interneuronal connections in accordance with the information derived from the target organs. And this is hypothesis. Okay. Well, and officially in Western country, the founder of this area of neuroplasticity is considered it is believed that it was defined by the Lugano, Italian psychiatrist. The Italian psychiatrist Lugano introduced the term plasticity in neuroscience. It is believed all over the world. People do not know Bechterew. They know Pavlov only because he was a winner of Nobel Prize, and the first one in medicine and physiology. Well, and he knew that throughout life, the anatomical functional relations between neurons can be changed adaptively to provide mental maturation, learning, and new functional recovery after brain damage. Well, but look, please, but all these, I will debate in a couple of minutes. All of these observations, neurological observations, at that time, in the 19th century, were controversial to fundamental ideas of the function of the brain. First, it was believed over this time that neurons are not reproducing. Then it was believed that neuronal networks and synapses are stable in time and space.
Sorry, we have 1 minute left.
Sorry?
We have 1 minute left.
Well, so individual neurons can synthesize only one, but it will leave.
That was not the
Individual.
What?
Well, be polite anyway, huh?
It's true.
Well, individual neurons can synthesize only one neurotransmitter. And the implementation of the genetic program and functioning of individual neurons is strictly predominant. Well, so next lecture, I will explain you that all these dogmas were completely reversed over recent 30, 50 years. Thank you very much.
Thank you.
See you in the seminar. This is a compensation for this strange bill. Thank you.